Yeah, I want to thank the CMU1 and all the organizers for doing a great job and inviting me for this long residency. Yes, today I want to talk about broadly uncovering hidden causes across disciplines. And I recently moved to the Medical University of Vienna, and I'm hiring PhD postdocs and interns, so spread the word. Yeah, and this is supposed to be a high-level overview of the research I have done in the last decade. And during that time, I have been fortunate enough to do research literally across all scales, from the smallest possible to the largest. So the smallest possible scale listens to the name, the Planck scale. It's the scale at which quantum fluctuations in the metric of space-time become so volatile that the notions of space and time lose their meaning. So there cannot be a smaller one. In one of my papers, we were doing calculations at that scale in the context of theories of quantum gravity. And by using some generalized notions of symmetry that we call dualities, specifically the gauge gravity duality, we were able to conclude about quantities that happen in conformal field theories, so completely different theories that don't even have gravity. And in those theories, we aim to classify those theories. And this is a very interesting task because those theories describe phase transitions. For instance, how magnets get magnetized, or how neural networks become from untrained state to the trained one. Yeah, and at the other extreme of the largest that have been used in science has shown up in the context of trying to resolve Hawking's quantum information paradox. Because in the last 8 years, people have realized that in order to resolve this paradox, you need to consider not just the universe in isolation, but consider possibilities of many baby universes, an infinite number of them, interacting by merging together or splitting up. And only when you take into account those interactions, you substantively move towards resolving Hawking's information paradox. Yeah, and both of those. And so in this part, we created the first, to my knowledge, the first rigorous 2D topological quantum gravity model with non-trivial non-gravitational sectors, in which we moved towards resolving Hawking's paradox. And in that sector wasn't like, so that non-gravitational sector could be in fact any orbit-closed Atiyah topological quantum field theory. But then during my postdoc, I moved and studied the middle scales, and in particular the cell, tissue, and organ scales, and created machine learning and generative AI to study several different problems across those scales. And one interesting observation is that, yeah, the theories that were required, at least in my research, to study those two extremes were quantum in nature, whereas here more classical theories were applicable. And also from a causal inference perspective, we see that there are some differences of how we deal with trying to figure out how one thing causes another in those scales versus the others. So in causal inference, we use a lot of DAGs, and causes are edges, but in these other two scales, we use more Feynman diagrams, and the causes are actually the nodes, not the edges. The edges are the variables that might be interacting. So sometimes there's different ways of thinking about things. So yes, in this regime, causality is about whether things are in the light cone with one another, and whether correlators are connected or disconnected, and whether you have those factorizations. Yeah, so like I was saying, in the middle I was using classical theories, whereas in the extremes, quantum theories. And what's the main difference? Well, the difference is that in classical theories, when you have two mutually exclusive possibilities, for instance, a cat being dead or alive, you cannot have both of them simultaneously. That's where the name mutually exclusive comes from, because you cannot have them, from the classical mindset, you cannot have them simultaneously. But in quantum mechanics, you introduce a new notion, the notion of superposition, which does allow for classically mutually exclusive possibilities to coexist at the same time. And that's where everything starts, actually. In In quantum gravity, of course, we don't deal with superpositions of cats. Gravity being the study of the geometry of space-time and its distortions means that quantum gravity is about superpositions of mutually exclusive possibilities for the geometry of space-time. And exactly, when you want to do a calculation in quantum gravity, for instance, a space-time that has 3 boundaries, you need to consider all the possibilities in between. That's what this cloud is supposed to stand for. So you need to consider all the different topologies that it could have. You see here some examples. After you fix the topology, you need to consider different geometries. And after you fix also the geometry, you need to consider different matter configurations that could be described by some extra theory on them, and consider all of those possibilities. That is what we did 5 years ago. And one of the theories, I said we consider all of the open-closed ATIA topological quantum field theories, but one example was the Chern-Simons theories, which have been used to study different exotic materials that, for example, like the fractional quantum Hall effect, and also predict new ones. But they were not invented for that. Actually, they were invented to solve knot theory problems. They were invented by Edward Witten to answer a challenge by Michael Atiyah regarding knot theory. And what they are, those theories are ways of computing new topological invariants of manifolds. The way you compute them is you essentially count how many principal G bundles you can have over a manifold. So you just count them, but then there's gauge transformations that you can use, so some of them are equivalent, so you actually count the isomorphism classes of them. And you might think that you need to weigh them by how big the classes are, but that wouldn't give a topological invariant. You need to weigh them by the inverse of that. And we did the calculation for those theories, and we get a generalized version of Mednick's formula. So here it's for any manifold with genus G and n punctures and conjugacy classes of the group G specified on the boundaries. And you get here a summation over the E reps of the group G, and which involves also the character of Q. So that is tantamount to taking into account all the possibilities for the principal bundle, because we literally counted all of them, we considered all of them. Then you need to sum over the topologies, which would be summing over the genus, and that would give you the connected correlators. For instance, it would give you when one of them literally splits into 2 and goes into the other ones. But other than that, there's also the disconnected correlators. For instance, this one moving towards there, and this one springing out of nothing. So the disk string diagram from like category theory perspective. Yes, and so after you've computed the connected ones, you need to sum still over the disconnected possibilities for the manifolds. And what that gives you is, what that gave us is this answer. However, the answer should have been, so here we have n3 boundaries, so we should have had the same theory, the same boundary theory 3 times. We should have had something that's raised to the n-cube power, but here this is not raised to the n-cube power, it's because it has this weird summation. So we didn't get one prediction. What we appear to be getting is a distribution for the values. So the boundary interpretation, the holographic interpretation of this theory, is that on the boundary you have a distribution. You have a distribution of quantum theories. So in other words, this 2D model is equivalent to one-dimensional theories without gravity, in which you are agnostic about the representation, let's say. More specifically, the Hilbert space of those theories is a representation that has all the E-reps, and each E-rep is in the number of copies that is a random variable that follows a Poisson distribution. But reading this statement in reverse tells us that if you have one-dimensional theories and you are agnostic about their parameters, that ends up literally creating new space and time, and in fact non-static space and time, because it also has gravity. This phenomenon is a phenomenon that biologists and biostatisticians also encounter every day, because it's an example of an emergent property, something that didn't exist and that arises out of more fundamental notions than itself. In this case, space and time arise from summations over representations of G. Yes, so yes, so this is the connection exactly between my PhD work and my postdoc and right now work, that I'm using those mathematical tools that I developed to study physical space-time to now study biological phenomena, because the way we have Feynman diagrams and we try to decompose a correlation of four particles that we observe into the different mechanisms that could have contributed to it, this is extremely analogous to trying to do that for genes, because if you have two transcription factors that are correlated with some downstream genes, transcription factors are some proteins that haven't been encoded by a gene. So a gene encodes a protein, and this protein forces another gene to be expressed. It's a form of gene interaction. But not all genes are transcription factors. So you observe a correlation between them, and you don't know how exactly did they directly cause the other two to be expressed, or did they cause a mediating one to cause them to be expressed? Yeah, so this is what, like I was trying to say, those tools from physical space-time are now applicable to biological space-time, because now we have the technologies to profile biology across scales in both space and time. For instance, here we see a recent paper where they were able to profile the cardiac muscle, both at the millimeter scale, physiologically, and at the same time at the micrometer scale, where you see individual cells. And now we have the technologies to know how much each one of our 20,000 genes is expressed in every one of those cells that you see, and also retain their relative locations. And similarly in time, by having those technologies in a high-throughput fashion, we're able to profile several time points along an organism, or several time points along the differentiation trajectory of a cell, of a group of cells. And what can we do with those very powerful data sets? Well, what I want to do is create generative AI-powered virtual biological systems. So simulate virtual biological systems in silico. And the holy grail for those, perhaps, is the so-called digital twins. The idea should be familiar, like I'm creating a digital version of, say, myself, such that if I get a disease, I can simulate it in my digital twin, and then and then try all the different treatment regimes on my digital one first, and only pick the best one to give to myself. But to do that at single cell resolution, this is too ambitious, hasn't been done for all I know. So instead of that, people have tried to do it in smaller biological systems. For instance, individual cells. You might have heard those models the last couple of years being referred to as virtual cell models. Yeah, as virtual cell models. And you can think of those two as different extremes on a dimensionality and scale axis. Well, the digital twins are three-dimensional because you have profiled the entire three-dimensional microenvironment of every cell, whereas those ones are, since I said they're dissociated, they literally take the tissue, they break it up in cells, and then they sequence every cell individually in this case. So they're zero-dimensional because you didn't sequence any of the other cells. You lost the information of what's close to what. But those models are very popular. There are plenty of those, specifically by the Charles Zuckerberg Institute. But there was pretty much a gap in between. So here, this is the relevant to the perturb-seq data. So those virtual cell models are trying to condense all the information from the perturb-seq data. But the paradox was that there is the equivalent of those genetic screens that exist in perturb-seq in the spatial context, but nobody had created those perturbation models of virtual tissues and organs, and nobody took advantage of those ones until, yeah, so I've created a couple of models in the dissociated regime, and I tried to fill in the gap for using spatial genetic screens and using them to train models and validate them. So that led to this other work, cell common. And what I'm interested in is moving towards the virtual whole organs direction. This is a collaboration with people from Cambridge, UCL, and San Diego. Sarah Teichmann, Peter Lee, Sandro, Claire, Swan, and Bolden. And yes, try to do that at the whole organ level at single cell resolution. Yes, so that's what I want to now zoom in, like try to explain the ones I briefly showed, but at each one level, one at a time. So yeah, so starting with empty drops. So this tries to solve the first problem you encounter in single cell biology. And that problem is that, so the way we sequence cells is that to do it in a high throughput way, we try to capture them in a droplet, which droplet will barcode all the RNA molecules in it. And then they'll release those molecules and put them in a soup, and then high throughput sequence the entire soup. And then you'll retrieve which cell they came from because of the barcode. That's the technique to do it high throughput. So the whole trick is to capture the cell in the droplet. But sometimes you try to capture it and you miss. Instead of capturing a cell, you capture just water with some RNA molecules because of the buffer where the cells were suspended. So then you get some RNA molecules, but you don't know if they were cell or just random noise. And doing this cell calling, deciding, can be very problematic because then you might start thinking you have some new type of cell, although it's pure noise. So we tried to solve this, and there was only one algorithm, the cell arranger arc, that was doing it based on, very crudely, based on the number of RNAs. They were like, oh, if you capture a cell, it should have a lot more RNA molecules. So let's decide that these are a hard cutoff in the number of RNAs. But of course, if the signal to noise ratio starts to be similar, for instance, a cell type that has few RNAs that are comparable to the noise level, then this approach is either going to have too many false negatives or too many false positives. So this way we develop a better approach that looks at each cell individually. And then you see here that in this sample, cell arranger arc missed half the cells. And what's worse, some cell types, like the oocytes, it missed to more than 80% because of this hard cell calling that they did. And we also applied this method to a cartilage development data set that allowed us to profile new cell types to an unprecedented level of detail. Yeah. Right, sorry, I was glossing over it. It's not generative AI, so it's not very sexy. So the way we did it is we like, so the way we did it, we're like, oh, take all the droplets and pick the ones, subset to the ones that have less than, let's say, 100 counts. Because those are so small that, of course, they're not cells. So then take them, pseudo bulk them, as we call it. So like add them all together, like you have a cells by genes matrix, like sum over the columns so that you have like one cell per genes. And that creates a profile of the soup. And then you do a statistical hypothesis test. So yeah, and then you do a statistical hypothesis test of every other droplet, whether it's compatible with this. So we assume that it follows a Dirichlet multinomial distribution, and then based on that, we were comparing, doing a statistical hypothesis test between the soup that we profiled and every other droplet. And if it was, the p-value was too small, so very unlikely that it comes from sampling from this noise, we were calling it a cell. Yeah.
Can you intentionally create negative controls?
Of course, we did simulations. Yeah, maybe given the crowd, maybe I should have presented more about this. But yeah, we also did a simulation because, of course, what I just described, you can simulate it, right? So we did a lot of simulations to show that cell arranger, of course, will miss it, whereas we would find it. And it was really striking plots where you see that, yeah, cell arranger completely misses a blob of cells, whereas empty drops multiome was able to single them out of the noise. Yeah, imagine that, that this one was true cells and are next to the orange ones. So Cell Ranger would miss both of them, but we would find that this is and this isn't. Yeah. Yeah, but moving on to cell dissect. So we made this cell dissect model. So this is one of the virtual cell models. And what we are trying to do here is a model that does end-to-end integration and perturbation for multi-batch and multi-covariate datasets. Because now, since the technology are getting better and better, we can now keep track also a lot of metadata for the cells. And yeah, one of the ideas we had was most perturbation models during training, they focus on reconstructing the observed counts. And then at test time, like I was saying, they were trying to do perturbations with them. But our idea was in cell dissect was that, if you're interested in perturbation time, don't only train using don't only train to reconstruct biology, but train to also do those perturbations. And indeed, we see that this is one approach that in population studies improves our predictions at test time. The overall architecture looks like this. So it's a mixture of the expert VEs, where each VE focuses on a specific covariate. For instance, the tissue the cell came from, or the age, or the sex, and then you can do perturbations. That's a method that scales very well in doing perturbations even to multiple covariates. And yeah, so the idea behind it is roughly this diagram, where every time we're marginalizing out one of the latent variables, and then we have a VE for each one of those. On top of that, we use some auxiliary networks to achieve better, to minimize the information shared between the mutual information shared between those latents. Yes, and then we devise different counterfactual scenarios to benchmark the predictions of the models, specifically on how well it can understand gender effects by making a lung immune cell from male to female in a specific out of distribution, in a context that makes it out of distribution. Also tissue effects, but also combined double tissue effects. And we see that we improve over two other famous methods for disentanglement in single cell data. Yes, so in machine learning, what we mean by disentanglement is having a latent representation of an object into a vector whose coordinates have different semantic meaning. For instance, the first coordinate of this could be that it is a cat, the second one about the color, and so on. And if you make a human visualization of the latent spaces we find in this simple data set, I think you can get how this latent space has come to learn a lot about the cell type, but not a lot about the condition, whether it was controlled or simulated, whereas the other latent space has the opposite, has a lot of mutual information regarding this, but very little regarding that. This is supposed to be more for visualization. However, then applying this model to a 9-organ immune atlas of the developing immune system, we see that it finds a latent representation of the data that splits what those authors had annotated as early megakaryocytes into two. Megakaryocytes are this stem cell that differentiates into the platelets, the cells that clot our blood when we are injured. But we see here that it splits it. Our model thinks that actually it's two different things. And indeed, if you look at this subset, we see that it expresses some transcription factors related to mast cells. So it seems to have some immune properties and some also unique genes. So it seems to be a different subpopulation of megakaryocytes that has immune properties. Yes, and now moving on to the next level of organization, the two-dimensional one, I want to talk about this self-common model, which is, like I was saying, to my knowledge, the first perturbation model of virtual tissues, in the sense of being the first to have been trained and validated on spatial genetic screens. So the task is of figuring out how one gene influences another gene within the cells, but also how one gene influences another gene in the neighboring cells. So the way we made it is we picked some well-motivated biological axioms, and then we processed them. Then we rewrote those axioms to reveal what the architecture of the model should be. It turns out to be a form of generative graph neural network, GNN. And yes, so the idea is that during training, you give it spatial transcriptomics data, and you let it learn those intra and intercellular gene interaction matrices. So basically, you give a cell, and you have a tentative causality diagram of what influences what in mind, and you find an embedding of the cell, and then try to predict the transcriptome. And if your prediction is very wrong, you'll have a big loss function, and then you have to back propagate to reduce the loss function, so make a better prediction and repeat. And that would allow you to go then and say that, oh, you know what, I'm gonna pick this cell and this gene and knock it out, and prompt it like that, and then ask the model, oh, how will that cell I change, and also the entire tissue change because of this. Yes, and in order to validate it, we looked, for instance, at human glioblastoma data. So we trained the model, and then we knocked out the interferon pathway. And yeah, we picked this random cell, and we knocked out the interferon pathway. And what should happen, like, if we think about it, like, the interferon pathway is the way our cells let their neighbors know that there was an infection. So if one cell doesn't let its neighbors know, then those neighbors also won't let their neighbors know. So if we knock it out in one cell, it should also go down in the neighbors and their neighbors. And indeed, this is what we observe there. But so it's giving in this situation some biologically sensible prediction. But of course, the very good criterion would be to do it in vivo, and to do it using spatial genetic screens in vivo. And fortunately, there was one that had been published, back then just one. And this was in a mouse lung cancer model where they had some wild type cancer and potentially one of two gene knockouts. So what we did is we trained the model on the wild type parts and to understand cancer biology. And then we picked one of the wild type spots and we perturbed it in silico and then compared this one to the ones that were actually perturbed in vivo. So this is a zero-shot prediction in the spatial genes, so it's a very hard task. But we see that the predictions have good correlations given how hard the task is. Yes, and then moving on to the third ladder of the discussion, we have this vision with of the collaborators that I mentioned, and also Andrew at UCL, of having those patient-derived virtual whole organs. This is like a very future perspective, in the sense, so those patient-derived organs, I'm using this term in relation to, you probably haven't heard of those, but there's those patient-derived organoids. So there's organoids that can be IPC-derived, in which, like, we study the general population, or they can be the organoids of my liver. So take samples from my liver and try to reduce my liver, not just a generic one. And they call those patient-derived organoids. And I'm appropriating the term patient-derived to mean virtual whole organs that are indeed tailored to the patient. because yeah, if we use low resolution 3D clinical imaging, of which there's plenty, we can use the other modality in our data sets, which is a super high resolution 3D imaging of the whole organ, and correlate the two, and then use this high resolution as a bridge to the molecular modality in our data set, to impute the gene expression and have the entire organ molecularly captured in silico. Because then if we use that data set and train generative AI models on it, then we would have those virtual whole organs I described that we can perturb in silico and then propagate predictions all the way back to the patient. So right now we're focusing on this part of the pipeline in two or three different organs, but correlating it to what is clinically available would be super important. Yeah, and on that note, I want to thank you all for listening and for inviting me here. Yeah, and if you have any questions.